special episode because this is technically my 20th full-length lesson video. I know technically this number says it's number 21, but episode 2 was just a short video with Slytherin as a guest star. It wasn't an actual lesson. So this is actually my 20th science lesson taught from home. So yay, I've reached a milestone. So today we're going to continue talking about conservation, and we're going to start by refreshing our memory on what conservation is. Hopefully you remembered that conservation is the careful management of Earth's living and non-living resources. So today we're going to take a look at successes and failures of conservation. We're going to look at an example of where scientists were able to jump in and help to bring a species back from the brink of extinction. And we're going to also look at an example where we acted a little bit too late and we weren't able to make things better. So before we get into this, I want to just go over some vocabulary, some things that kind of explain what we're talking about. So a couple of weeks ago in one of the videos, I mentioned about this thing called the red list. And this is a list of all plants and animals, pretty much all plants and animals on this planet, and it uses different rating categories, which we see here, to help us understand how much that organism is at risk of extinction. It doesn't just base it on the total number that exists, it's also based on what kinds of threats is it facing, it's based on how quickly does that animal reproduce, when it does reproduce, how many offspring does it have. So there's a lot that goes into these categories that's not just based on the total number of individuals on the planet. So if you see here in red, these are the categories that are of most concerned. I should also mention just before I go on, you can move up and down this list. So for example, bald eagles in the United States used to be considered endangered, but they had a lot of success in stopping destruction of their habitat and of other problems that were affecting them, and they are not endangered anymore. They've moved up the list. Vice versa, polar bears, some of you may know, over the past couple of years, they have dropped down the list. They used to be near threatened. Right now they're moving into vulnerable. It won't be long before their status officially changes to endangered. So organisms can constantly move up and down this list depending on what's happening to their population and to the threats they're facing in the wild. Also, before we get into my example, I should mention that for conservation, we tend to use what is called charismatic megafauna. Oh, that is a big college level word there. So what this means is, in order to get people to care and to donate money and to be concerned about saving plants and animals, it means that we often use the cute and the cuddly stuff. Charismatic means it's cute, it's cuddly, it's exciting. Megafauna means it's big, right? So that's why when we talk about conservation, you tend to see images of pandas because they're adorable. You see images of cheetahs or tigers or lions, all these really exciting animals. But in reality, there's a lot that's out there that is at risk of extinction that's not cute, it's not cuddly, and it's not something you and I may be aware of. So the World Wildlife Fund has been working on campaigns to bring awareness to the fact that there is more than just pandas out there who are endangered. So this advertisement is very clever. There are these fish, which are pretty not that exciting, all kind of gray, but this fish is wearing a mask of a panda, and the caption says, "Would you care more if I was a panda?" Because the fish that is in this picture is endangered just like the panda. And I'm not going to tell you right now what the fish is. I will leave that as a question on the self-quiz, and we'll see if any of you get it right before I reveal the answer. But there is definitely a very large fish in the sea that we are eating at alarming rates that is putting it as endangered just like pandas. And yet, they're not very exciting fish, so they don't typically get their own billboards or their own stuffed animals, and they don't tend to get the attention that a lot of other organisms do. So because of this, I am purposely not going to talk about a charismatic megafauna. I'm not going to talk about the panda. I'm not going to talk about the tiger. I'm not going to talk about cheetahs. I have purposely decided to pick an organism who's maybe not the prettiest looking, and has been one that has got an amazing story behind it. So before we get to that organism and its story, the way that conservation works is scientists are essentially doing an experiment, right? We see an organism that is at risk of extinction, and in order to stop that from happening, we basically need to do an experiment to see if our idea for how we can save this species will happen. So I bring this up because this is how the scientific method applies. Oh my gosh, we're going back to the scientific method, because in order to do conservation, in order to save animals from extinction, oh my gosh, we have to use the scientific method, just like we were doing an experiment about paper airplanes, or maybe we were doing an experiment about baking soda and vinegar. We need the scientific method. So hopefully you remember that the first step of the scientific method is form a question. The second step of the scientific method is form a hypothesis. The third step is to design an experiment. The fourth step is to collect and interpret data. And finally, at the very end, we form a conclusion to wrap everything up. So I am going to use the scientific method while I talk about these conservation successes and failures. So I'm going to identify what is the problem or the question. What did scientists think? What was their hypothesis? What were the materials and procedure they used? So I am going to explain my examples using the scientific method as my reference, because the scientific method is how we get stuff done, and it's how we make sure that we are approaching things in a very systematic way. Alright, so what is the organism we're going to talk about? We're going to talk about the California condor. Again, not the most maybe attractive organism out there, but California condor is very important, not just as a namesake bird for California, but an important part of ecosystems. So let me start by just giving you some facts, and then we'll go into the conservation story. So what ended up happening was by 1981, and 1981 was not that long ago, in 1981 there were only 22 birds left in the wild, and that's in the wild in California, in Arizona, in Mexico. There were no more California condors. There were only 22. Why this happened was through a lot of different reasons. One of them is from condors being poisoned from lead in bullets. Condors are scavengers, so they will eat dead carcasses, and if those carcasses have bullets in them, they'll ingest the lead. There is also some research to suggest that a chemical called DDT may have affected them like other birds in weakening their eggshells. And also, a lot of California was obviously going through huge development, same with Arizona and Mexico, so their habitat was also being destroyed. So, however we got there, the situation was 1981, 22 birds left. So the question that scientists had was, could the California condor be brought back from the brink of extinction? Could we do something to not only stop those 22 birds from potentially dying, but could we do something to get more birds, to have more California condors back in the wild? So, as in the scientific method, scientists needed to figure out what they were going to do. So they came up with a hypothesis. And their hypothesis was that moving all of the currently living condors that were in the wild, and then collecting any remaining eggs, taking those out of the wild, that would help to stop the extinction of the California condor. So the scientists decided, right, to make a major gamble here, right? Because they thought if they just left those 22 birds in the wild, they would end up becoming extinct. So instead, their hypothesis was, hey, we think collecting all the birds, collecting all the remaining eggs, and bringing them out of the wild, that is going to be the way to stop them from going extinct. So how did they design this experiment? Well, the San Diego Zoo Global, they were given permission by the U.S. Fish and Wildlife Service and from the California Department of Fish and Game to begin the first zoological propagation program for California condors when there were only 22 of the birds left in the world. So basically, San Diego Zoo and then later the Los Angeles Zoo, they were given permission that they were allowed to collect birds and collect eggs in order to build a breeding program. So in order to do this, what did they need as materials? Well, they needed to have a special, and they actually called it a condominium. Get it? Condominium. And it basically had these very large free flight enclosures. They actually had 6 of them, and it would still allow the birds to fly and cover long distances, but they definitely had, nets over the top of them to prevent them from escaping. They also needed a bunch of incubators for the eggs. They needed meat, which is what the condors eat, so that meant rat and mice and rabbits, which they obviously had to raise at the zoo in order to feed them. They also needed a lot of black clothing and covering for incubation chambers, and we'll see about this in a moment. And they also needed adult condor puppets. Wait, what? Why would they need puppets? Well, they ended up coming up with a pretty cool idea. So in 1983, they started collecting eggs, and as these eggs were hatched, right, and they put them in the incubators, they needed to figure out what to do next. So this here is a picture of Sisquawk. He's the very first California condor who has ever hatched in a zoo. He came out of his shell on March 30th, 1983, and he was kind of the first hope that this program could be successful. But they wanted to make sure that Sisquawk, as well as all of the other baby condors who would be hatched, they wanted to make sure that they never got used to humans, right? Because some of you may know, if humans raise baby birds or other baby animals, then that animal thinks that the person is its mommy, and then they, have a harder time being returned to the wild. So instead, what the San Diego Zoo did is they created these puppets, which you can see here in the picture. So basically, the vet or the zoo technician, they'd stick their hand inside of the puppet, and that is how they interacted with the bird. They made sure that the birds never ever saw or came in contact with people. So they would actually play tapes of adult condor sounds, and several times a day, right, the condor puppet would come into the incubator. It would stroke the baby bird just like the mom and dad would do. The puppet would have food inside of its mouth. You can see that here in this picture. And they would feed the baby bird. So they made sure that every single condor chick that hatched, that they only had contact with a puppet, and then later, once they got a little bit bigger and they weren't in any danger, they were then introduced to other adult condors and put into an enclosure with them. So the main idea here was to make sure they never came in contact with human beings. They got the idea from in South America, that working with Andean condors, they had used this idea, so they were able to make this work. I should also mention that another thing that they did to help increase the number of condors. Condor parents typically only raise 1 egg at a time, but if the first egg that they lay, if that egg gets destroyed, like it gets eaten by a predator, or it falls out of the nest, or maybe the parents accidentally step on it, the mother bird can very quickly lay another egg. So what they did is, when the birds in the zoo would lay an egg, the keepers would come in and they'd steal the egg, and then the parents, right, they would lay another egg, and so basically they were able to double the number of eggs that were being laid by being a little sneaky and stealing the first egg that the birds laid. As a result of this, in 1987, the last condor was brought into the wild. He was called AC9. He was the very last condor that was caught, and A really high point was reached when Moloko, who was the first zoo-bred condor, hatched on April 29th, 1988. So as the years went by and they started building up the population, they eventually got to release. So in 1992, the first zoo-bred condors were released into native habitat in the Los Angeles National Forest. And this was kind of the first step to see if these birds could survive on their own. As of today, what is kind of the data, the collecting and interpreting data? So in the early 1990s, we started releasing birds in the wild. And today they're released at 5 different sites. There's one at northern Arizona that's just north of the Grand Canyon. There's one in Big Sur at Ventana Wilderness. There's one at nearby Pinnacles National Park, or Monument. It used to be National Monument, but now it's National Park. And there's also one in the Los Padres National Forest. So the important part here is if we do an experiment, we need to start monitoring it, right? We need to start collecting data that it's successful. Well, before we release them, we obviously stick some stuff on them. We use what is called radio and satellite GPS telemetry. So that's basically achieved by putting tags on the bird. They obviously have tags on them that give them a number so we know who they are, but they also have like a small computer chip embedded in them that sends out satellite and radio information, and that way we can pick them up with a receiver. We also use old-fashioned binoculars and telescopes, right? Just simply having researchers go out to the Grand Canyon or out into Pinnacles National Park and look for the birds. And then we've also got some cool remote sensing weather stations that have been set up, especially in a place like Pinnacles National Park. It not only is tracking the weather, and then we can use that information to study how weather affects their behavior and their flight patterns. Back in the zoos for a moment, I mentioned Sisquawk. He was the very first bird that was born in San Diego Zoo. Well, he has a mate named Satash, and they are often featured on the Condor Cam raising their chicks. Sisquawk has gone on to become quite a daddy and has done a good job of taking care of future children. And so 165 California condors have hatched at the zoo since Sisquawk arrived there in 1983. And you can definitely check out, if you go to the San Diego Zoo or the San Diego Zoo Safari Park, the Condor Cam is still running because obviously the zoo is still open, not for visitors, but to take care of the animals there. So you can definitely check out the California Condor Cam and check out what the birds are up to. So the good news is the California condor is still critically endangered, but it's doing a lot better. As of October 31st, 2004, there were 425 condors in California, and of those 219 were living in the wild. So the conclusion we can draw from the experiment that was done is that captive breeding of California condors using the San Diego Zoo methods was successful, right? Incubating the eggs, using the puppets to raise them, later letting them have access to adult birds, and then later releasing them, we have brought the California condor back from the brink of extinction. So the program is still continuing though. Why? Because the main threats come from environmental toxins, especially lead. They also can sometimes run into power lines, especially because remember these birds, you know, did not see power lines in captivity. They can also be shot, and obviously there still is a bit of a collection happening with museums, egg collection that often happens by people who are doing so illegally. They still are in trouble. So that is why they're still critically endangered, because they still face some threats in their environment and from people, and the numbers of them just aren't high enough yet to take them off the critically endangered list. And what's important about this story is that this really laid the groundwork for lots of other conservation efforts for a variety of other animals as well as birds. So back in the 1980s, when this started to become successful, other scientists realized that this same technique could be used with a variety of other animals, and it really helped to build confidence in programs that started with captive breeding. And in fact, this book by Jane Goodall, it's called Hope for Animals and Their World. It basically, every chapter is about a different organism that has been brought back from the brink of extinction, or they're currently working to do conservation work. And so they talk about in this book that the California condor, what they did, that same technique was used for things like crested ibis or whooping cranes. So there is a lot of conservation being done, and a lot of that is made possible because of the early successes they had with the California Condor program. So now we're going to look at a not so happy example. We're going to take a look at where the conservationists were not able to help quickly enough. So I'm going to warn you right now, this next story does not have a happy ending. So the next organism we're going to talk about is the Pyrenean Ibex, also known locally as the bucardo. So there's all kinds of different ibexes throughout the world, but this particular type lived in the Pyrenees Mountains in parts of Spain and France. And this ibex was incredibly well adapted for this environment, having to deal with climbing extremely high and sheer cliffs, also dealing with very cold temperatures at times. But it's really awesome antlers that, if you guys remember, the males used to fight with each other to gain access to females. Well, those horns were exciting trophies. So they were hunted quite a lot, and by 1913, they were thought to essentially be extinct in the wild because of so much hunting that had happened. But in 1913, that very same year they were thought to be extinct, they did find a small population in a kind of secluded valley of the Pyrenees, and they only had about less than 100 individuals in that population. But it looked like there might be a sign of hope. So in 1913, the question was asked, could the Pyrenean Ibex be saved from extinction? So here, scientists need to make a decision. They need to make a hypothesis. They need to make a guess about how they can best save the organism from extinction. So the hypothesis that they ended up coming up with is they thought that a ban on hunting, so passing a law and creating a national park, would be enough to save the Pyrenean Ibex from extinction. So in order to do this, their designing an experiment was pretty simple. They established a national park and boundaries of that park. They created a national law to ban hunting, and later on, not in 1913, but later on in about like the 1980s, when technology started getting better, they also started taking tissue samples of ibexes that were alive so they could start analyzing them and figuring out, exactly how closely related this ibex was to other types of ibexes around the world. Well, in 1989, only about 6 to 14 bucardos or Pyrenean ibexes were still alive inside the park. In 1996, 2 of the last 3 remaining animals died, so that means there was only 1 left. And unfortunately, that remaining Pyrenean Ibex, named Celia, she died in January 2000. Her skull was actually crushed by a fallen tree. Oh, it's like horrible. It wasn't even Celia's fault. It wasn't a human who did it. There was a really bad storm. She happened to be sleeping under a tree. The tree fell and it crushed her skull and she died. So yeah, that's a failure. The Pyrenean Ibex, the experiment to try and conserve them and bring them back from the brink of extinction, it totally failed. So the Pyrenean Ibex is no more, it is extinct. Or is it? So this is where the story gets a little bit science fiction-y, but it's not fiction, it's fact. So in 2009, some scientists came up with this idea that let's clone a Pyrenean Ibex, because if you remember, they took tissue samples of the ibexes and they had them in, deep storage, and so they were still viable. So this gets a little bit complicated, but basically they took the tissue sample and they took the nucleus out of that cell. And remember, the nucleus is important because that's where the DNA is. And then they took an egg from another ibex species. They took the nucleus out of that and they basically put the empty egg cell with the new nucleus inside of it, and then they added a bunch of hormones, they got it to start dividing, and then this bundle of cells, which is what we sometimes call a blastocyst, they put that into a surrogate mother. Now, they obviously couldn't use a Pyrenean Ibex because they don't exist anymore, but they used other types of existing ibexes, and I believe they had about 30 different surrogate mothers. Unfortunately, only one of those pregnancies actually went to full term, that it didn't miscarry, and in 2009, a Pyrenean Ibex was born. This here is the baby that was born. Sadly though, it only lived for about 7 minutes or 10 minutes. Some of the papers I've read say 7 minutes, some of them say 10 minutes, but basically the Pyrenean Ibex came back from extinction for like 7 minutes, and then it died. So this cloning attempt was definitely not successful. They also tried again in 2014, but none of the pregnancies carried to term, no babies were born. So obviously the story of the Pyrenean Ibex shows what can happen when we don't take aggressive enough measures to conserve animals. And it also brings up the very controversial science fiction-esque topic of, could we use our advances in genetic technology to start basically cloning organisms and bringing them back from extinction? And I will include in Google Classroom a link to some interesting articles about this because it's a very controversial subject, but also an area of curiosity, right? Just because we can do it, should we do it? So we've now looked at some successes and failures of conservation. These were just 2 examples for today. Tomorrow we'll look at 2 more organisms, both of which I will tell you. One is a definite success. One of them is a work in progress, but definitely still headed more towards a success category. So we'll have both happy stories tomorrow. Make sure to check out the self-quiz in Google Classroom. And also, if you're interested, you can explore a little bit more about cloning extinct animals.